Yeah. So what you're seeing here is a picture actually of a new institute that we've just launched in Cardiff, which I'm the director. It's called the European Cancer Stem Cell Research Institute, which is a very, very long name, but I'll explain a little bit about the idea behind that in a moment. It's based in a new £26 million building, which we got the university here in Cardiff to invest in. And they've also invested another £3.5 million in supporting our activities within that institute. So it's a very exciting new activity within Cardiff, expanding the research base. So this next slide just shows my group, or part of my group, actually on a walk trying to raise some money for Tenovus, in fact, that we did over the summer in the Lake District. And you can see on the right-hand side, actually we've got a sizable group composed of a number of postdocs, a number of PhD students, and some technical support as well. And they all work within the new institute. And highlighted just on amongst the students there in red are Meera, Shami, and Stephanie, who are the 3 PhD students who are currently funded by Tenovus. Meera is going to tell you a little bit about her project, which has just come to an end, and she's about to move on to go up to an institute in Glasgow. Shami is in the middle of her PhD, and Stephanie is just commencing her PhD last week, so we've got the full range of PhD students funded by Tenovus within the group, for which we are very grateful. So we don't do our work in isolation. This is just to give a feel for those of you not familiar with the situation in Cardiff of all the many, many different structures which fund cancer research within Cardiff. So we're very fortunate to receive funding from a number of different organisations, which include, of course, Tenovus. And they all interact. And actually, Tenovus feeds into quite a number of different activities within the School of Biosciences, within the medical school, and indeed within the new institute. So it supports a range of activities across Cardiff from a range of different funders with a range of different objectives. So my group and the institute itself is kind of interested in a number of different questions, which I'll sort of very briefly introduce them to you, or a number of different problems, shall we say. The first problem is shown here, and you won't be able to see it, it's too small, but essentially what this shows is the drug development pipeline. And what it shows is how terrible we are at making new drugs to cancer. On average, it takes, to deliver one new drug into the clinic, into patients, it takes us about $1 billion US, okay? 500 million pounds, to go from the process of first identification of a possible target through to a drug in a patient, 500 million. It's also incredibly wasteful. You start out with about 10,000 compounds, and at the end of it, you arrive with your one compound, which is going to be effective. It's very, very difficult to make new drugs. And part of the reason we think it's difficult is actually we don't think the way that we look at drug development is as efficient as it could be. And maybe we need better models and better platforms to tell us whether our drugs work earlier in the pipeline where it's cheaper to lose drugs, so that we can actually have a more efficient drug delivery method. So that's one problem. The next problem is that actually for some cancers we really don't yet understand the basic nature of the cancer. So what you're looking at here is, if you can see it, is survival rate. You don't need to see the detail on the slide. The take-home message is that survival rate, 5-year survival rate for some cancers is very good, but for others it's abysmal. So for example, for pancreatic cancer, we simply don't understand the disease. We don't know how to treat it at the moment. And that's probably because we don't fully appreciate how the disease develops, we don't understand the genetics behind the disease, and we don't have proper targets for it. So we need a better basic understanding of disease processes for some cancers. And the third problem is relapse. And what this shows you, again, you probably can't see it from the back there, but this shows you relapse rates for breast cancer. So these are patients that are cured, and this is the frequency at which their breast cancer will come back. And in fact, the frequency over 15 years, for example, after you've been cured, is about 15% of breast cancer. So the question is, did we really cure the breast cancer? Or did we just put it into abeyance for a while so that it will then come back and regrow? That's one question. The other possibility, of course, is just that those particular patients are predisposed to getting breast cancer, and actually the relapse is a new breast cancer. But that question has actually given rise to one of the concepts that we're trying to address within the Institute, and that's whether actually all cells within a cancer are actually not equivalent. Indeed, we know that's the case. If you look at a cancer carefully, it's quite clear that all the cells within the cancer are not the same. And the question we want to ask is whether some cells are actually the driving cells within the cancer. And in fact, most of the cancer is just bulk cells, which are not actually important to the growth of the cancer. And actually, it's those driving cells that we should be targeting with therapy rather than the whole bulk of the cancer. And this notion is called the cancer stem cell concept. And it's the idea that a very small proportion of the cancer is actually responsible for the cancer's growth and its relapse. And if we understood that concept a little bit better, this is our idea, we'd have a better set of targets to go after in terms of druggability, to repress not only the cancer when it presents, but also the possibility that it will relapse. So that's one question. Sorry, so druggability is the capacity to develop a drug that will hit your target and make the cancer shrink. And to do that, obviously, as I've said already, we've got a significant investment to push that effort forward. The other thing that we're interested in, and this is directly talk to what Meera is going to talk about in a moment, is can we actually make therapy personal? We know that different tumors of the same types, for example, breast cancers, will present with different genetics. And the question is, can we use the different genetics to actually tailor therapies to individuals? So not all breast cancers are the same, for example, and can we select drugs that will specifically target particular types of breast cancer and get those to regress? So that's tailoring therapy to make therapy better. And in fact, Meera is going to talk directly to that issue in terms of drug development. So these are the areas that my group focuses on: intestinal cancer, prostate cancer, and breast cancer. It tends to be the epithelial cancers, skin tissue cancers that we work on. And the sort of tools we use material from patients, which we are now growing out in what we call organoid cultures, and that means we grow mini breasts, mini intestines out in culture, which we can then test therapeutically. And we also use mice. So we use the capacity to genetically modify mice to ask precise genetic questions about genes which drive cancer and genes which may drive cancer regression. And I'm going to show you a very, very brief movie now to show you the sorts of things we can do. This is the intestine, the small intestine. And as we zoom into the small intestine, you'll see it's composed of finger-like projections. That's what does the absorption, the main function of the gut. Okay, so it's the absorption of nutrients. So these finger-like projections are hang out into the lumen of the gut, and they are supplied with cells from what we call crypts. And the crypts here are where the stem cells sit. And the stem cell is the key cell that drives the growth of the whole structure and maintains the whole structure. And what we can do with some genetic tricks is we can introduce labels into any cell actually we want to. And in this case, what we're doing is we're introducing a colored label into the stem cell of the intestine. And then we can actually just watch that stem cell rebuild the intestine. And that's what you're seeing here, is that stem cell at the base of the crypt is building the whole of the intestine over a period of 3 days. And you and I, our intestine turns over, the whole of your intestine turns over in 7 days. In a mouse, it turns over in 3 days. So what you're seeing here is that process. And because we can play with these genetic tools, we can actually identify the cells that drive the growth of the whole structure. Okay? So we can find the stem cell. And this is the first time you can really do this sort of approach in vivo to look at, that means in living tissues, to look at what really is the stem cell. So the next trick you can play is you can ask where cancer originates. And one of the hypotheses we've been testing is whether actually the stem cell itself, that one cell at the base of the crypt, at the base of this structure, is actually the cell that's important for tumorigenesis. So what we can do is we can, again, play some clever genetic tools, and we can either start the cancer process within the stem cell, or we can start it outside of the stem cell. And we can ask the question, which is more efficient? And so if you start the answer to that question is going to be that if you start it in the stem cell, it's incredibly efficient. The stem cell is the origin of cancer. So what we can show you here is the crypt. And what we're going to do now, into that stem cell at the base there, we introduce a mutation which drives cancer. Just one mutation, it drives cancer. And then we watch how that cell should build the intestine. And it starts off normally, and it's building away there, the green color, but then something goes wrong. And this conveyor belt of cells, which normally take the cells out into the lumen of the gut, stops working. The cells start to invaginate, move into the surrounding tissue, and there's your cancer. So in a period of a few hours, we can watch from initiating that mutation into the stem cell, we can watch that stem cell turn into the origin of a tumor. So that's the sort of technology we can use, and we can play with that sort of technology to ask a number of different questions. Sorry. So the sorts of things we're trying to get at with those sort of approaches are really listed here. So sort of better understanding of the fundamentals of cancer, what drives it, building better models of disease so we can find new therapeutic targets that don't exist already, but also make that drug pipeline more efficient so that we don't waste so much money and we don't spend so much time in developing new drugs. So at that point, I'm going to hand over to Meera, who's going to tell you about her PhD.
Thank you. Right, so a bit of science now. I apologise if it's too confusing, but so my project has been looking at developing personalised therapies for specific types of bowel cancers. So bowel is an essential organ in the human body, as you can see here, and it's critical for our digestive system, and it's where we absorb nutrients and water, as Alan has mentioned, and it's also where we generate our waste. So therefore, the development of cancer in this organ has debilitating consequences on the human body, and this actually affects about 16,000 people every year in the UK. So although over the last sort of 30 or 40 years, the mortality rates of bowel cancer have been declining, the incidence rates have actually been increasing, suggesting that there's a continual necessity to develop new drugs and new ways to combat and treat this disease. So the way that we're currently treating bowel cancer is regularly using surgery to remove the primary tumour, together with chemotherapy and radiotherapy. So the issue with these two methods is that they're quite toxic to patients. So they act by killing cells which are rapidly dividing. This is a property of cancer cells, but the thing with this is that they also target normal cells which rapidly divide, such as your hair follicle cells, your digestive tract cells, and also your bone marrow cells. And this is what leads to the unpleasant side effects experienced by patients, such as nausea and hair loss. The way that we'd like to treat cancer is obviously by better prevention and screening methods to identify the tumours at an earlier stage, but we'd also like to be able to personalise the treatment and be able to treat the individual type of tumour, as Alan mentioned previously, according to the genetics of the tumour. So together with being able to use less of the nasty therapies, we'd like to essentially deliver a treatment regimen which effectively treats the cancer and has very low side effects for patients. So the way that we can do this, have I gone the wrong way? Oh, no, sorry. Is by understanding the genetic mutations within the tumour to then direct the therapy to those particular mutations. So, as Alan mentioned, the normal intestinal epithelium is maintained by a number of proteins, and it's mutations in these specific proteins which leads to the development of cancer. So, for example, mutation in the APC gene, which he spoke to you about earlier, leads to the formation of small set of neoplastic cells, and these are precancerous cells. And then it's mutations in other proteins, such as KRAS and PI3K, which lead to the formation of metastatic disease, which is then able to invade through the bowel wall and invade other organs via the bloodstream. So essentially, information about proteins such as these gives us, from information such as proteins like this, we're able to tailor our drugs according to proteins such as KRAS and PI3K. So these are known as biological or targeted therapies, and essentially these are therapies which are specifically designed against well-established targets, and these are critical for driving tumour growth. So therefore, these treatments will hopefully target only the cancer cells and spare the normal cells in the body. So an example of this is cetuximab. Some of you may have heard of this. And this agent is an antibody which is used to target a protein known as the epidermal growth factor receptor. So this protein is present in normal cells, but it's also found to be increased in cancer cells in about 70% of bowel cancers. And the function of this protein is to activate other proteins such as KRAS and PI3K, which I mentioned previously, and these proteins then drive growth of the tumours. So what this antibody does, I don't know if you can see the little two-pronged thing, is prevent the actions of this epidermal growth factor receptor and prevent growth of the tumour. So this is translated into a clinical benefit for patients, as you can see by the survival curve on the right-hand side. So from that, you can see that 100% of patients are alive at the beginning. I don't really have a pointer, but so with time, you can see that the patients that received cetuximab had an increased median survival compared to the patients that didn't have cetuximab and only had standard of care treatment. So although this treatment shows some benefit for patients, unfortunately, this is only available for patients that don't have mutations in KRAS or PI3K. And this is because even in the presence of this antibody, the mutations to the damage to the protein will continue to drive growth of the tumour. So the aim of my project was to evaluate some new therapies for cancers which have KRAS and PI3 kinase mutations. So for this, we employed the use of models which mimic the human disease, so in terms of the way that the tumour looks and also the genetics that the tumours have. And we were also able to obtain drugs which are not in the clinical pipeline for bowel cancer at the moment. But so we have one drug which targets proteins following a PI3K mutation, and another drug which targets proteins following a KRAS mutation. So from our studies, what we found was that the use of the drug which targeted proteins following a KRAS mutation resulted in a significant increase in survival in our cancer models. And similarly, we saw this was the case with the PI3K targeted drug following a PI3 kinase mutation. What we were also interested in was tumours that have mutations in both proteins, and this is because about a third of all bowel cancers have mutations in these two proteins. So what we found was that with individual drugs, this doesn't translate to a survival benefit, unfortunately. And we think that this is potentially because the presence of the other mutation is driving tumour growth in these cases. So what we did find was that the use of both agents together in a combination treatment led to a synergistic effect. So the two agents synergized and improved survival, as you can see here. So what our studies have shown is that there are drugs which are available for these tumour subtypes. And very excitingly, we're very excited about the start of the MRC Focus 4 trial earlier on this year in Oxford. And what this study is hoping to do is to look at a large patient population and identify patients with KRAS or PI3K mutations and give them drugs, similar to the drugs that we've been using, to hopefully find a patient population that will respond better to such treatments. So I wanted to take this opportunity to say thank you to Tenovus for sponsoring my PhD. and also for sponsoring me to attend conferences all around the world. So this was really useful in going and talking to other scientists and discussing research, and also visiting other laboratories. So I was able to go to San Francisco to the American Association of Cancer Research Conference, and also to Barcelona for the European Association of Cancer Research Conference, and also Liverpool. I don't know whether you believe that that's a picture of Liverpool in the bottom left, but it is, to the NCRI conference in my first year. So to sort of give back, Alan, as he mentioned previously, put us all forward for the 10 Peaks Challenge earlier this year. There's actually 10 of us from the lab. It's Kirsty and Lee sat in the audience as well. And we actually thought this was going to be quite enjoyable at the start, and then at the end, as you can see, I'm barely able to stay awake. But we raised over £2,000 for Tenovus, and we just wanted to say thank you for sponsoring research in our lab.